let's get started with the features. We have two new features in this release. I'll go to ToxoDB. One is a new chip-seq workflow that is in our Galaxy instance. So I'm up here on my workspace, analyze my Galaxy data, and then from I guess I have to log in. And I will end up on the home page of our VEuPathDB Galaxy instance. If I scroll down this middle section here, which is our, essentially our home page, I can see that we have a new chip-seq workflow. If you do that type of work and want to look at your data privately on our Galaxy instance, you can use this workflow. So if you click on that link there, it just takes you to the workflow, and you can, if you have data in a history, you can easily run the workflow. So yeah, if you use that, click contact us and let us know how it goes for you. We would love to hear it. And our second feature that we have is a way to use the basket within a strategy to look at certain subsets of data. So it's like a basket strategy interplay. And I'm going to take some time here, maybe about 5 minutes, to go through this feature. So first, I'm just going to quickly create a two-step strategy. So the first one, I'm going to demonstrate a new RNA Seq data set, this monensin treatment in sensitive and resistant E. tenella strains. It has biological replicates, so we can look at our statistical analysis. And I want to compare the resistant cells incubated for 2 hours in monensin versus the sensitive incubated for 2 hours. And I'll look for upregulated genes that were differentially expressed by a 5-fold or better with a p-value of 0.01. And if I click Get Answer, I find that there's 170 genes that are upregulated in the resistant cell line. And then, I'm in the strategy system, and we have all kinds of data you can look at. And let's just add a transmembrane domain. See how many of those monensin genes have transmembrane domains. And I can select all of these and just run. So the search returned 50,000 or so genes with transmembrane domains, and 26 of them are at the intersection of this RNA-seq data set. So 26 of them are upregulated in the resistant cell lines and have transmembrane domains. So, this morning I was looking at some pathways and some genes, and I saved 7 genes in my basket. These are the ones that I'm working on in the lab and I'm really interested in. And so I'm interested to know whether these 7 genes are in the list of upregulated resistance genes. So if I focus back on this in my strategy on the first step, I see that I have this new, let me make that bigger. I have this new feature here. It's called Show Only Genes in My Basket. And if I turn that on, then this table is filtered by the genes in my basket. Everything that is not in my basket then is hidden from this table. And I see that I actually only have 3 genes here, and there were 7 genes in my basket. And that's because not all the genes in my basket were in this list of 117 genes. So I'm going to turn that off again, and we'll look at the whole table. The other use of this feature is that it can be used to view search-specific data columns for other steps in your strategy. And search-specific data columns are data that only apply to a specific data set. So for this RNA-seq analysis, you know, we produced fold change and other information that it really only applies to this data set. And I can show you that. So if I go to add columns, I have fold change and adjusted p-value for this data set. But if I look at these 26 genes and the data available there, I don't have those columns to turn on in this table. But with this new feature, this new basket feature, I can filter this by the 26 genes in my basket to be able to look at those columns for these 26 genes. So let's go ahead and do that. I have these 26 genes here, and I can say send to my basket, and I choose to replace the 7 genes in my basket with these 26 genes. I'm going to click that. And I get this green light here that says my basket is ready. If I refocus on my resistance genes, I can say show only the genes in my basket. And now I have a list of 26 genes. They're all in my basket. They have this little green basket icon. And if I go to add columns, then I can turn on fold change and adjusted p-value. It was actually already turned on for that. And so I can look at this data specifically for these genes that I'm really interested in. And that is the end of the new features. I will hand it over to Eve then, and she is going to talk about the new data in FungiDB.
All right, thank you, Suzanne, very much. Let me switch the screen over to me really quickly. Okay, you should be able to see now the FungiDB webpage with a news release for release 64. Okay, so I will walk you briefly through the data sets that we have integrated in this release, and I will start by reviewing the genomic sequence information. So in this particular release, and I'll make the page a little bit bigger so you can see it better, we have a number of new genomes, and the organisms that are integrated are of medical and veterinary, industrial, and also agricultural significance, and they cover a number of different organisms of interest. In this release, we also have genome replacements for 3 genomes in total. And the genome replacements use long read sequencing that was used to improve genome assemblies. So we used new genome assemblies and replaced the original genomes that were in FungiDB. So all of the 3 genomes for Histoplasma capsulatum are improved chromosomal label genome assemblies, and Histoplasma capsulatum is a human fungal pathogen that causes fungal respiratory disease in immunocompetent hosts. The 3 genomes represented here belong to North American, African, and also Panama clades, and span 3 different lineages that are quite interesting in terms of geographical and also SNP distribution. Right, we also have a number of transcriptomics data in this release, and the data spans established or emerging human pathogens such as Candida auris, Cryptococcus, Sporothrix, and Telleromyces. We have also data for model organisms such as Neurospora crassa or Podospora, and agriculturally important pathogens such as Bloomeria and animal pathogens Pneumocystis. So notice that the data sets that are listed in each of the categories are also linked in blue. And these links will redirect you to the dataset record page. So if I click on the link for the Telleromyces marnifae, it will redirect me to a dataset record page for this particular data. And so the data record page contains different information. At the top, you'll have information about the title of the dataset, the type of the dataset that you're looking at, such as RNA-seq, the organism that was used the organism that was used to align the reads from the RNA-seq, such as the main reference genome for this organism, and also a little bit information about the samples, how they were treated, whether there are any replicates, and main contact information for the providers. So within the same page, if you scroll down a little bit further, you'll have access to various links that cover how you can explore this dataset within FungiDB, link outs to data in NCBI, and also graphical representation of one of the genes that can be studied within this dataset. So if you were interested in examining these datasets, you could click on, let's say, a fold change and look for upregulated genes. And so first, what we are looking at here is to set up a search parameters, and I will look for genes that are upregulated, and let's say I'll switch it to the fold 2. I have selected the reference sample mycelia and the comparison sample as Arthoconidia. So Telleromyces is a pathogen that can exist in mold and yeast form. So it is endemic to Southeast Asia and can cause opportunistic infections in immunocompromised hosts, but it is thermally dimorphic, meaning that at upregulated temperatures, it produces conidia that are also the infectious component. And so you may be interested in looking at this organism and determining the genes that are upregulated in the infectious stage of this organism. So let's go ahead and click on Get Answer, and we will return essentially all genes that are corresponding to our selection criteria. Notice that you return quite a few genes. On the bottom, the list, there's a list of the results, such as genes that are upregulated by 2-fold or more. And on the right, you also have graphical representation of how this gene behaves. So we expect to see this gene or that gene to be upregulated within the conidia samples. Now you could also at any point change the parameters of your search. For example, if you click on Edit and go to the Revise, and now you say, okay, so 1700 genes is quite a bit, and I want to return actually highly upregulated genes. So what you could do is change the fold change to 5 and look for genes that are upregulated by 5-fold or more. So that also provides a more stringent criteria for looking for highly upregulated genes. And notice that we now have 437 genes that you may want to take a look. You can also analyze these lists further in order to gain additional information by clicking on the Analyze Results tab, and for example, examining these lists via gene ontology enrichment process or metabolic pathway enrichment, or even looking at the word enrichment, seeing if there's possibly a number of product descriptions that are overrepresented in the 400 genes that you have. So this is quickly how you can analyze the transcriptomic data, integrate in FungiDB, but obviously you can add more steps to bring in any additional types of data that we have. So now I'll briefly come back to the news on FungiDB and tell you a little bit more about other data sets. So in addition to transcriptomics data, we have also integrated whole genome sequencing of isolates and various strains. And in this release, we include data for Aspergillus fumigatus that causes aspergillosis in humans. And there are 2 data sets associated with clinical isolates and those that also have been evolved in the lab. We also have 17 clinical data sets for Candida auris, which is an emerging and I guess by now probably already established human pathogen of medical importance and concern globally. And for these data sets, the 17 isolates were collected from a Texas lab and also sequenced. And we have a number of isolates that were collected in Australia for a plant pathogen Fusarium verticillioides. Now, in addition to this data, we have also integrated genome browse tracks. Oh, and just before I cover, I want to step back and talk a little bit more about SNPs. SNPs can be examined via similar strategies, just like transcriptomics data. So if you click on the data set, you'll be redirected to a dataset record page with a list of available SNP searches. And just before the webinar, I ran a search for a dataset that uses Candida auris data. And I looked for non-synonymous SNPs, essentially, that may be of interest if you're thinking in terms of drug resistance phenotypes in Candida auris. And so here's my table with all of the genes that house non-synonymous SNPs. And I want to briefly demonstrate that in addition to examining the datasets via a dedicated search strategies, you can also view these records on gene record pages. For example, for the first gene in my table, I clicked on the link for the gene ID, and I'm now redirected to the dataset record page for this particular gene. So gene record page contains a number of resources that may be useful to you when you're interrogating the function of the gene. And in this particular case, if I click on the SNPs, I can now visualize the SNPs that were calculated not only from the dataset that we integrate in this release, such as non-synonymous SNPs that I mapped, but also from other datasets that are currently integrated in FungiDB. So this is a very nice way for you to look at the SNPs that may be similar between different datasets globally using the integrated data provided in FungiDB. Right. And now just quickly touch base on the genome browser tracks that we have. This is a genome reannotation project where authors used RNA Seq and also manual and computational approach with Maker software to reannotate genome and possibly discover novel genes that have not been previously annotated. We're providing this data set as a genome browser track where genes such as dark red or blue are the genes that match the description, such as genes in terms of the classes, and the additional annotations such as CDS or accents are provided in a lighter shaded tones. And so for this, in terms of FungiDB, I think this is all from me, and I will pass it back to Suzanne or Omar.
Great, I'll take it over from here. Yep. Okay, so hi everybody, and again, just as a reminder, feel free to ask questions at any time. So there is a question panel in GoToWebinar where you can type your question. If you really feel like you want to ask it verbally, just raise your hand, and hopefully we'll have some time and we'll unmute you. But otherwise, if you type it in this way, it ensures that we get your question and we can answer it. So I'm going to focus on 3 databases. And in particular, I will focus on our integration of single cell RNA Seq data. So we'll be looking at HostDB and PlasmoDB, and in those databases, we're actually going to be focusing on the data from the same study. So this was a data set where the investigators infected mice with Plasmodium burkii, and then looked at the expression pattern of host and parasite genes in infected cells, in infected liver cells. And they also did some analysis to determine also the structural location of those cells within the liver. So we'll do that in a second, and then we also, so that's HostDB, PlasmoDB, and what I'm showing you here are just the news sections. And as you saw before, here I'm jumping to ToxoDB, and as you can see here, I can click on the news section again, and I can go to release 64, and there's a news for ToxoDB. And Suzanne already showed you this new data set in ToxoDB, which is this RNA-seq data set. Eimeria is obviously, it's not a toxoplasma, but it is a related avicomplexa, and it's available for evolutionary comparative purposes in ToxoDB. So there is also single cell data in here. If we have time, I will show you this data as well, but I will most likely focus on host and PlasmoDB for the Plasmodium data set. I just wanted to make sure to highlight one thing, and sorry, Suzanne, if you're going to highlight this later on again, but it doesn't hurt to remind people. But we did also integrate updated functional annotations. So those are the names of the genes and their product descriptions for Toxoplasma gondii MA49. And this was an update to 803 genes. And so this is a big, significant update to functional annotation. And that's obviously largely driven by the community. And we're looking forward to also updating the structure of genes in an upcoming release based on community input. And if you're interested in knowing how you can contribute your annotation, I encourage you to go to our tutorial section and look for tutorials that cover Apollo annotation, and or entering user comments on our gene pages, depending on how you want to enter that your information and annotation. And so if you search this page for Apollo, you'll find here that there at least one webinar that was based on Apollo annotation, but there are additional ones as well. Okay, so I'm going to jump back to HostDB. And so HostDB is a database, one of our resources that its goal really is to integrate host response to a pathogen infection. And so, you know, of course, as we know, most of the parasites we're interested in, or organisms we're interested in, infect hosts. You know, and in our case, we're interested in infection of mammalian hosts. And often what you're, at least in HostDB, if you're interested in infection of vectors, obviously you would look in vector-based. But the bottom line is that you can come in here and start asking questions about changes in host gene expression, or changes in host protein expression, or other characteristics based in response to an infection. And so in this case, the data set we're interested in is this data set from Anhai Afayat et al. And in this experiment, if we go ahead and click on this, you'll get the data set description page, and you will notice that for this data set description page, we have a section called Associated Datasets, and you'll notice here that there is there are a couple of associated datasets that are in PlasmoDB, right? So this tells you that, okay, this data set the study has more than one data set in it, basically. And so while here we'll be looking at focusing on host response data, you could focus only on the parasite genes if you're interested in looking at that, or looking at the parasite side of things. That said, the data we'll be looking at today in HostDB does include the parasite genes as well. So both are combined in there, but you can filter them out as well if you're interested in. For all of our data sets, we have a section called Explore this data set in this website. So you could, straight from the data set description page, you can go and go straight to asking questions about this data, and we'll do that in a second. But importantly, you can also go to the paper from this page. So you'll see here there's associated publication, and if you click on this, it'll open up the link to the paper. And then, obviously you can, and that's obviously we highly encourage you to do that, is that if you're interested in querying and analyzing this data, it's good obviously to always look at the paper and understand what they did in the paper, so you make sure you're asking or you're using the data appropriately. But importantly, I think it's an important thing to get in the habit of doing when you're looking at databases, and that is to make sure that, well, do the results look right? Am I getting results that are similar in the paper, or is it way off? And so you can make sure that what you're that you're confident about the data that's available to you in a database. Okay, so I'm going to go back here, and I'll demonstrate one quick search, and then we'll dive into looking at the data in a special app for exploring single cell data. So we have a search in all of our databases if single cell data is available, where you can identify genes based on single cell data. So if I click on this, you will notice here that this data set is selected. It's the only one in HostDB right now, so that's the only choice you have. And it's an experiment that was done in mice, and this is the experiment. And so I can just click on Get Answer. And what this is going to do is just going to give me back a list of genes that were detected in the cells that were assayed in this experiment. So this is not terribly informative. It allows you to subset basically results. So for example, if you had, as you saw with Suzanne, where she had a two-step strategy, I could start off with a list of genes that I'm interested in, and then ask, well, were any of these genes also expressed in this single cell experiment? And then you would go in and explore those further using an app that's tailored to exploring single cell data. And so I'm going to go back to the news section. And looking at the explore section, you'll notice that we have another option here to explore this data set in cell by gene. And I'm going to click on this so it starts loading. This often takes a few seconds to load. So there's a lot of, the app has to load in your browser, and then it has to load actually the information that you're going to plot on the various graphs. So we'll give this a second to load. And there are different ways of accessing this cell by gene app. So one is through the dataset description page. The other way is when I ran my search. If I go to my strategies where I ran my search, you'll notice you automatically get these custom columns loaded here, where I can explore, go straight to looking at this specific gene in cell by gene to see where, so if you had a list of genes that you really want to just go straight to them, you can do it here, or you can explore all this data on the gene page, and we'll go to that in a minute. Let me double check and see if there are any questions so far. Don't see any. Okay. Okay. So what we're looking at here is a projection, basically a UMAP projection of the cells. So basically, in a very, very simplistic manner, the cells are clustered based on their expression pattern. So something, cells that are closer to each other have something similar to each other in terms of the expression profile of the genes in those cells. And so really, it's just a way to look at a large amount of data in a more simplified way. So in a 2D graph, basically. It's not telling you anything about the expression of the genes right now. This, where the dots are, doesn't tell you anything about the expression of the genes. It's just telling you that the cells that are closer to each other have something in common with each other based on their expression profile. And so you're basically reducing the large amount of data from the expression of all the genes in these cells, both host and parasite genes, right? Many of these are infected cells, into something that is that you can look at on a 2D space. So you're looking at this 2D space, but of course now these are all just black dots, and you're like, well, I don't know what this means, right? So that's where it's important to be able to color these dimensionally reduced graphs using available metadata or information about the samples that you analyzed, or the cells specifically. And you'll notice here on the left, cell by gene has all the metadata that's available or that was provided to us by the people who generated this data. And you can see that there are different categories. So for example, there's something called infected tag. I can expand this and you'll see here there's a false and true. You can also, you'll see these little droplet icons. These are the color icons. So if I click on this, you'll notice here it's showing me which ones were uninfected. These that are false, the purple ones, and which ones were infected, the green ones. And so you can quickly see that, okay, most of the cells were uninfected, right? Which is not surprising from an infection study. And you notice here there are many different types of metadata you can sort, you can color on. So for example, you can color on by time point post-infection, right? And so you can see here now, you can see where the infected cells were after, the clusters of infected cells based on the time point of infection, and then not infected are right here. And then you can also obviously remove some of these. If you only want to look at a handful of them, you can do that. And so that's easy to do that here. And yeah, so that's basically the bottom line here on the left side, is that you can really color and visualize your data in a way that's correlating with the metadata that you have in for these samples or the cells. Okay, so another thing you can do is I'm going to go ahead and go back to the paper here, and the first thing I was looking at, I was wondering, well, which genes are upregulated in infected cells, and they go into detail here, talking about how they found these genes that are involved in iron chelation that are upregulated, and so I'm going to just take one of these genes, FTL1, and go back to cell by gene, and right here at the top I can paste that ID in here, or that gene name in here, and you'll notice FTL shows up here. I can click on this. Now I filter to this particular gene. I can expand this. You can see the expression profile of this gene with the number of cells, and I can color based on this gene, right? So now I can look and see where this gene is expressed. And so now if I go ahead and, for example, take out, these are the infected cells, and you can see that these are infected cells that have high expression of FTL1. I can even, click on this graph here on the upper right-hand side, and I can really focus only on places where there's maybe a higher upregulation, for example. And so I can really tailor this to whatever I want, and then you can detect the cells where this gene was highly expressed, and you can correlate it with whether this were infected or uninfected based on the various criteria. And also you can look at obviously the time point of infection and explore that as well. So the other thing you can do, I'm going to go back to PlasmoDB, and I'm just going to look, or sorry, not Plasmo, I'm going to go back to HostDB, sorry, and I'm going to look for FTL1. So this is the FTL1 gene, and you notice I got 4 results here. So I'm using the site search option up here. So I got 4 results, and you'll notice that these 4 results are found in both mice and rats, but I'm only interested in mice. So I'm going to filter based on mice and apply, and here's the mouse gene. So I can go to the mouse gene page. And so again, as Eve was showing you, in FungiDB, you can look at the mouse, at the fungal gene page with a lot of information. Same thing with HostDB, you can look at what information we have, and you'll notice there's a section here for transcriptomics. You can scroll down if you want slowly through this page, or use the table right here, or you can just simply click on this transcriptomics section. So I'm going to show you 2 things. So this transcriptomic is a summary of all the RNA sequence data that's in HostDB, not only Plasmodium infected cells. Remember, these are any host cells that we have with different infections, spanning fungi and protozoan parasites. If I scroll down a little bit more, you'll get all of the graphs associated with different experiments. I'm going to collapse these, but you'll notice there is a section for single cell data. And now if I open this up, it's going to load the cell by gene app within here. It doesn't have the full functionality. It's just a UMAP plot, and you can then click on this. And so this is showing you the expression of this gene in the cells in this in the UMAP plot. But obviously, if you really want to delve into it, going into cell by gene is the way to go. Now, remember, this gene was highlighted as a gene that's upregulated in Plasmodium-infected liver cells, and they had some rationale that maybe this being able to get rid of iron, which is important for Plasmodium in the liver, is a defense mechanism by the host. And so I was interested in knowing, well, are there any other infection models where this gene FTL1 is also differentially regulated? And I started by looking at this summary of RNA sequence data set at the top. Each one of these rows is a different experiment. And then this bar plot or whisker plot or whatever you want to call this plot here is basically every dot is a sample from this experiment. And so you can see what is a dynamic range of expression between the samples in this experiment. So you can very quickly see that, oh, this one down here has a large dynamic range. There's samples that are really far apart from each other. So maybe this is an interesting one to look at. So you can click on it. And this is a transcriptome from mouse cells infected with toxoplasma. And you can see here, they looked at different mouse cells, and you'll see that the difference is between uninfected and infected astrocytes and other cell types that are infected or uninfected, right? So it seems to be there's a cell type differentiation in the upregulation of this gene, and maybe not necessarily related to toxoplasma infection. The point here is that you can use this to start seeing if there are any correlations between infected cells and between different infection models from different organisms. So here's looking at another one, looking at, again, brain infection and toxoplasma, and you'll see that these type 3 bystander cells seem to have an upregulation of this chelator gene compared to uninfected cells, right? So this may be an interesting experiment to look at, and you might start identifying some correlations between different infection models. Here's one with Cryptococcus, but again, the lung cells, whether they're infected or uninfected, had a higher expression of this gene than brain cells infected or uninfected. So that's of course of interest. Anyway, the idea behind this is that this allows you to explore the data and start coming up with some potentially interesting hypotheses that you may want to explore further. So I don't want to take too much more time, but I will quickly jump to PlasmoDB, highlight that we've loaded a couple of new genomes and annotations. So it's always nice when you see these high quality assemblies that are being loaded with their annotation, because that then allows you to start looking for potentially missing genes or structural anomalies that were present in the older annotations based on the older technologies. We've loaded some genetic variation data, some Plasmodium vivax results from patient isolates. And then you notice here's the spatiotemporal data sets again. So I can quickly go to this experiment, and I'm going to, so this is host and parasite. I'm going to go back. I think I picked the wrong one. Oh, parasite only, that's what I want. And so let's go in here, and I'm going to quickly just explore this data in cell by gene. So I'll jump straight to cell by gene. I want to show you one more feature, and then I'll turn it back to you. So again, this is from, these are only cells that are infected, and they filtered out, you can see they only showed the true. And it's again a dimensionally reduced projection. If we go back and look at the paper, and you scroll down to, here, so this looks similar to this basically. This is basically replicating what's in figure 4. It's never going to look, I mean, this looks pretty good actually, but often you don't necessarily have to get the identical projection. And so what I can do here is I can, for example, take time points post-infection, and I can color this by the time of infection, and you can see that this is the 12-hour time point, 24, 2 hours, 30, 36. But what I can do is I can take and click and drag around here, and I can select a section right here, for example, these cells, and I can click up here to add them to my comparison panel up here. And now I can select another set of cells. Let's say I'll pick these cells right here, and I'm going to pick them as cell group number 2. And now I can click on this differential expression button up here. And so what this does now generates, finds me all the genes that were differentially expressed between when I compared these two groups to each other, right? And so now I can start looking at these, and I can say, oh wow, these genes are seem to be upregulated between these two samples. I can explore these further, look at them, again, highlight, do the same thing of highlighting and seeing where they're expressed. And of course, you can always go back to PlasmoDB with either this gene name or gene ID. We don't have control on what the provider or the people who generate data call these. Sometimes they use a gene name, sometimes they use a gene ID. But I can copy UIS4, and we can just search for UIS4. And again, you'll find it here, and I can just limit this by Bergii. And so there are 15 of them, and then can take a look at the genes, and then you can figure out, well, which one is it? And so I'm going to go with this first one because that's the gene symbol for that one. And now you can go ahead and take a look at it. And again, you can explore now the Plasmodium Bergii gene page and look at all the associated data and explore whether this gene is expressed at these different stages in other experiments and get some more evidence to support your data. So for example, I can take a look at this experiment here, which looks at the transcriptome in different liver stages, and you can see that this gene is upregulated in earlier infection time points, and then it goes down in different hepatocyte cell lines. And so if we go back to cell by gene, and I let me just do this, and so in theory, I should be able to see this, and you'll see here, I'm going to the highly expressed version of these. These were the earlier time points, if you remember, so I can color again by the time points, and you can see here, this is 12 hours right here. And so now if I color by this gene, you'll see that the cells that were infected for 12 hours have a higher expression of this particular gene. There's a lot more you can do with cell by gene, and of course, I didn't do justice to the paper itself. This paper they did a lot of work in here, looking at also the structural resolution of the liver and looking at where cells in the liver structure, which cells were infected, and differences between infected cells versus uninfected cells. So there's a lot of data that can be explored in this, and you can explore them using cell by gene. And as a reminder, using the metadata that's here on the left allows you then to subcluster and take a look at cell types of interest. So I'm going to stop here because we can go on obviously for hours just talking about cell by gene, but hopefully this gave you a flavor of what you can do in cell by gene. And Suzanne, turn it back to you to finish up the webinar.
Okay, it sounds great. Yeah, that was a great demonstration. Yeah, afraid my talks, I just want to go over and show you what data is in AmoebaDB, TrickDB, and VectorBase, and I won't have such a fantastic demonstration of data though. All right, so we're in AmoebaDB, and AmoebaDB received several genome sequences, so assemblies, and these appear for the first time in AmoebaDB. We have requested the annotation from the providers. They would need to upload that to an INSDC repository and meet those repository requirements before we could download them onto our site. But we've requested that, and if when that annotation appears in the repository, we'll be able to load it. But we still can take advantage of these genome sequences, right? These assemblies. And one of the major things that you can do with those is to blast against that. So if I don't know, go back to my ToxoDB data and get a sequence from one of these genes, say I'm really interested in this gene, I'm on a gene page here that was in the result of my Monensin treatment. If I go to sequences, I can get the genomic sequence part of that and go back to AmoebaDB and search the genome of all of these guys. Why not, for my sequence? and the job will run. I can do our BLAST tool is multi-BLAST capable, so I could search a couple different targets, probes at the same time. Sometimes these can take a little while to run, especially on the genome. But it eventually comes back with a list of areas in the genome that this applies to. And some of the hits have reasonable e-values and some of them don't, but you know, between very distant species, you might find something interesting. Okay, so I'll go back to, oh, the other thing that we integrated was a proteomics data set, and that would be really interesting. It's not very much, so to find the search for that, you could go to look for genes by proteomics and quantitative mass spec. So this came to us as a group of numbers. So we would have that under the quantitative data. We also integrated several proteomics data into TrickDB. I'll go to the news there. And we have 4 new data sets here, and the search is very, you know, similar to all of our other searches. Let's say I'm interested in what genes are secreted. This was actually a time course of gene secretion experiment where they incubated the Trichomonas vaginalis cells for up to 2 hours and looked at proteins that were left in the media. So I can look for those proteins by going to search for genes based on proteomics and quantitative mass spec. I have the dynamic secretome here, and we, because of the way the experiment was formatted and how the data came to us, we have the full change search that is available for that. And I can, let's say I'm interested in upregulated genes between the 60-minute control, the 60-minute control and the 60-minute 37-degree incubation. I can click Get Answer. And I find 344 or over 300 genes, and which of course has all of the associated data with it. We graph the data that's available in the column and also on the gene page in the proteomics section. And then the other advantage, of course, is that you're in the strategy system and you can add a step and combine that with any of our searches, data from any of our searches. And we run a lot of bioinformatic analyses on these data sets before they go in. So onto VectorBase. VectorBase actually received quite a bit of data this time around. I'll go to the news where it's listed. We integrated 3 annotated genomes that are new to VectorBase this time. And of course, that means that if you go to the BLAST page, you can not only search the genome for different data, you can search the transcripts and the proteins as well. So that's a really powerful. And if you also are interested in, as we integrate these, we run all of these different analyses and create searches. There's over 100 different searches against these annotated genomes. We have quite a bit of new RNA sequence data that we integrated. Most really are mosquito cell line data or tick cell line data. But the interesting thing about the way these are organized is that you'll be able to use a search strategy system to directly compare these cell lines. So let's say I'm interested in what is the difference between this C636 cell line versus the C710 cell line. I can, go to the dataset page, or I can just go directly to the search page and search for that search there. So C6. And I would like to know the difference between the C7 and C36. Say I'm doing an experiment and I want to choose these, what my model system will be. I can come here and look at what's the difference in the transcriptome of these. So I'm going to look for genes that are upregulated in the C710 at a fold difference of 2 and a p-value of 0.01. And I find 941 genes. So that's a pretty interesting thing to be able to do. I'll also go back to the news. There are also some of these data sets that are actually incubated with different infections. So you can explore what you need to do just by going to the search pages or by to the dataset record page. And then our pop bio data, we've updated 14 different projects, and they usually are just adding a little bit of data to each of these data sets. So if you are a pop bio data set, if you use our map view, you can, and one of these is your data set of interest, you can go to our map view. And search for that there and see what the differences are. I think that's all I wanted to show. I wonder if there are any questions.
I don't see any questions in here, but Stephen, who's on the call, sent an email to the helpline, which I saw about the order of the samples and the infection time points in the Cell by Gene app. And he's right. The order is weird. I think it's ordered as 12-hour, 24, then 2, and then 30, and then 36. And so I'm sure you'll get another response from the person who works on closely with Cell by Gene and integrating data. She might know better. My suspicion is often programmatically, these programs, how they sort things is they sort them by the first number in the 2-digit number. So 12 comes up first because it's 1, 2, right? And so 1 is first. And sometimes there's a way to override this, and if there is, we'll do that. If there isn't, unfortunately, we may be stuck with that until the people who develop Cell by Gene implement the fix. But yeah, that's a good observation. And unfortunately, right now we're stuck with whatever Cell by Gene allows us to do. But hopefully, there's a way for us to enforce an order on this.
Right. It actually
and I guess
things like that are actually
go ahead.
Much more difficult than you think to fix.
Sometimes they are, yeah, exactly. Although they should be straightforward. The other thing is to point out is as Stephen did, he sent an email to our helpline, and so maybe Suzanne, you can just point people to where the help, where people can click on the contact us link, and you'll see up in the upper right underneath the search bar, you'll see there's a contact us link, and you'll get this form, and you can just fill it up and provide your information, and we do our best to respond within 48 hours. And so you'll get a quick response. Hopefully we know the answer to your question. If not, maybe we can point you in the right direction.
Yeah, and no question is too small. Even if you're like, I think I used to know that this feature was here, but now I can't find it.
Right,
It might
exactly.
still be there,
exactly.
right?
Yep.
It might be a bug that it's missing. So you might be helping us out to keep our sites up to date.
Exactly. Cool.
Yep. All right, that's great. You should receive an email in a day or two with a recording of this, and you can always go look at the recording elsewhere too on our site.
Great. Thanks, everybody. Bye.
Yeah, thanks for coming.
Thanks.